So my name is Nick Pannunzio, and I'm an associate professor at the University of California, Irvine, and part of the Chao Family Comprehensive Cancer Center. What my lab really likes to study is genome instability and how that affects cancer outcomes. So we're really interested in the etiology of cancer. So exactly what is it that changes inside of a person's cell to lead to that to become a cancer? And my major focus is on blood cancers and heme malignancies. In particular, one cancer that we study now is a really aggressive form of acute lymphoblastic leukemia that highly impacts our Hispanic and Latino community in Orange County, which UC Irvine serves. So the CRLF2 mutation is typically associated with this subtype Ph-like ALL, which is actually a very recent subtype. So it's actually in 2015 got its own classification by the World Health Organization. And what it really shows is that the presence of this rearrangement and overexpression of the gene leads to patients having worse treatment outcomes and lower survivorship. And we see this predominantly in our Hispanic and Latino community, where it seems that a large number of the cases of Ph-like ALL affect this community. And among those, they have this mutation. And so if we could actually understand what it is about this population that makes them prone to this rearrangement, we would really start to understand risk and stratify risk across this population for this cancer. So one thing about blood cancers is they're usually defined by a certain type of recurrent chromosomal translocation. So this is when you usually have a chromosome that's intact and it breaks and it repairs by switching chromosome arms. And so this is the mechanism that's driving the CRLF2 overexpression. So what happens is that we have these normal processes in our B cells. And so B cells are the cells that are affected by this of VDJ recombinations. And that's what happens with this CRLF2 rearrangement is that something happens during VDJ where that double strand break isn't repaired correctly. It leads to this translocation with CRLF2, which essentially allows that cell to become immortal and is the first step in cancer for this Ph-like ALL disease. So one really interesting thing that we wanted to interrogate was that a lot of technologies exist to be able to analyze patient sequence data, but a lot of times it's very expensive and cost prohibitive and time consuming. So what we really looked into was these technologies involving digital PCR, which allows us to essentially look at mutations and quantify them in these distinct areas. And what that has really allowed us to do is to save time and money by running through lots and lots of these DNA samples to be able to detect mutations there. And what the hope is that this could actually become a clinical diagnostic tool in the future to be able to, track relapse of these certain types of cancers. So there's been a lot of large genomic data sets that have been out there. So TCGA is obviously one of the classic ones, but a lot of that is based on self-identified race and ethnicity. And what we really wanted to do was to convert a lot of that data to this genetic ancestry information. And so the advantages of something like this is that we can look at a population and see that people that maybe identify as Hispanic or Latino have this JAK2 mutation, for example, which leads to a more aggressive type of this type of cancer. What we can actually do with that now is essentially using the whole genome sequencing data, converting it to the from the self-identified race and ethnicity to whole genome sequencing, and actually now kind of bin those populations that have, say, a JAK2 mutation with certain genetic ancestries or admixture. The thing that's most exciting to me about this project is what will unlock a lot of potential and genomic sequencing data that's already out there or that people already have that they've done in their own labs. And so I think what's really exciting about that is not only the underrepresented admixed American population, but also the Middle Eastern North African population, which is actually absent in a lot of these studies, meaning that we don't even know some of the disparities that could be occurring in this population just because they're invisible in a lot of these large genomic data sets. At UCI, I mean, I feel like we really have this charge to serve our community. I mean, we actually, part of our cancer center at UCI has this Office of Outreach and Engagement. And what that allows us to do is actually go into our community and have events and kind of explain cancer risk and the benefits of participating in clinical trials and consenting to different studies. And so through mechanisms like this, we really try and engage our community to be able to realize the potential that they could have to help other people. It's really a terrible thing, when someone is suffering with cancer, and it's really a sacrifice for them to agree to be a part of these studies. And so I think it really means to us a lot as a community, but also I think it helps the greater good in order for us to be able to understand a lot of these different types of cancers much better.